Hello, welcome to the Pacific Postal Computing Association podcast. I'm one of your hosts, Derek Jacoby. I'm a PhD research manager at the University of Victoria and a director at PCCA. I also am involved in the How to Grow Almost Anything class, which is a MIT and Harvard production on synthetic biology that all of you might be interested in if you're into biology topics. We've got another class starting up in January, and look up HTGAA on Google, and I'll put the links in the show notes. My co-host, Piyush Acharya, is my co-author on this paper. And Piyush, do you want to introduce yourself?
Yeah, thanks for the intro, Derek. My name is Piyush Acharya, and I'm a rising senior in high school from Bellevue, Washington. And I've been working with Derek on various projects since 2023.
Excellent. Thanks, Piyush. So we're going to today talk about a paper called Mobius, a mixture of experts model for separating ME/CFS from long COVID. Both ME/CFS, or chronic fatigue syndrome, and long COVID are post-viral illnesses. So they're triggered after you get a virus and in the recovery to that virus. And from a symptomatic perspective, they're very easily confusable. In fact, doctors are able to separate the two illnesses only correctly about 58% of the time. And so from a symptomatic perspective, almost impossible to tell these apart. What our paper is doing is using epigenetics as a means to achieve this separation. Both of these illnesses are diagnoses of exclusion today. There is no good lab test. And previous iterations of epigenetic tests were getting accuracies only in the 80% or so kind of range. And we'll dive into those numbers a little bit later. But with our system, a deep learning system based on a transformer architecture and a mixture of experts, we're getting about a 97.1% separation, which hopefully is getting in that realm where we can start to think about this as the statistical basis for future lab tests to provide positive diagnoses. Piyush, do you want to talk a little bit about the introduction of the paper and possibly the data sets that we used in the paper?
Yeah. So for people who might not be super familiar with what our paper is and what its contributions are, let me outline that briefly. So first is our epigenomic classifier, and it's designed to differentiate between ME/CFS and long COVID and healthy controls. So that's the first contribution. The second is we've made some improvements to the transformer architecture, bringing it to a new state of the art for epigenetic data. And finally, we've open sourced our entire pipeline that includes the code, the model weights, and the process data sets as well. So with that in mind, I want to jump right into the data that we utilized. So epigenetic data comes in various forms, but we've chosen to focus on something called DNA methylation data, which is where methyl groups attach to DNA molecules. And these levels are recorded using widely available machines called the Illumina 450K and the Illumina 850K arrays. And what we've done is we've sourced data from the NIH's NCBI Gene Expression Omnibus, which is basically like a very big repository of data collected on these machines. So in total, we've got 14 distinct data sets comprising of 852 total samples. That's 284 ME/CFS, 284 long COVID, and 284 healthy controls. So that's the data that we're working with. Again, I've got 2 points to make here. First is that the data is all publicly available, meaning that like it's prone to batch effects, and the typical limitations of publicly available data apply. So we'll talk a little bit about this in the pre-processing step, but I just want to leave that there. And second is that all of the ME/CFS data sets come from before 2019, which means that those ME/CFS patients can't also have long COVID. So yeah, with that in mind, we've briefly outlined the data sets. Derek, anything to add on data, or should we move on to pre-processing?
Yeah, a couple things in there, I guess. Point about all the data sets prior to 2019 is really important because long COVID itself can be a trigger for ME/CFS. And this is why, since there is a little bit of a difference in underlying cause, it looks like that it's harder to separate things today. One of the future extensions will be to get some post 2019 data sets and see if we can still separate even from people who have had long COVID. But those data sets are a little bit harder to come by. They're coming in the pipeline. We should have an update on those eventually. Yeah. The other is in terms of your comment on batch effects, and I just wanted to clarify for folks in the audience a little bit about that. Because we've taken these from a bunch of different studies, about 14 different studies, their recruitment protocols were all just a little bit different. Their definitions were all just a little bit different, and they do fall into these broad categories of ME/CFS and long COVID, but we don't have quite as much control over the original data as if we had collected it ourselves. And so part of our pre-processing is really to enable us to get to the most reliable elements of that data.
Yeah, thanks for adding that, Derek. So yeah, why don't we just jump into some of the things we've done in that pre-processing pipeline? And again, the full list is going to be available in our paper, but I want to outline like the key steps that we've taken in that process. So
Yeah,
first
perfect.
is, yeah, so first we've fixed some of the batch effects of all these experiments being run with different protocols and different being run on different machines using a batch correction package in R called Combat. And we've also normalized all of the quantiles, so that's normalization and standardization, using a package called MinFi and Noob. And we've also then taken the top 1,280 sites based on a differential methylation analysis from that initial 450K. And just to clarify, we mentioned in the beginning that our data comes from two sources, right? Two machines, the Illumina 450K and the Illumina 850K. What we've done is we've distilled the 850K array data points down to 450K. So they're all even across both machines. And from that 450K, we've then conducted a differential methylation analysis using an empirical Bayesian strategy, and then taking the top 1,280 sites based on a statistical measure, and we then z-score normalized that, and then took the beta values, we put it onto a feature embedding, and that's how our CPG tokenization process works. So that brings us to the tokenization process and leading up to pre-training.
Yeah, that's great. So tell me a little bit more about how so we went from 850K on the large array. The step down to 450K was just choosing those CPG islands in common across the two chips, correct? And then it was
Mm-hmm.
from that 450K that we further distilled down to the feature set?
Yeah, yeah, and one thing to clarify here is that the 850K array that we both keep referring to is a newer version of the 450K, and because it's newer, it has more data points for more CPG sites, but it covers all of the data points that the 450K array covers, and so we're able to take the ones that are mutual between both machines. And
Yeah, good.
regarding like how the input to the transformer works is so typically LLMs are trained on tokens, right? But how do you deal with tokens when you have data about CPG islands? So for that, what we've done is we have divided each feature vector that we've created from those from the CPG sites, where each token, each feature vector has a total of 320 CPG sites. And so each embedding you can think of has 320 CPG sites, and that's one token, okay? And so the reason why we've done this, and Derek can explain a little bit on the biological side of why this works, but essentially CPG sites like move in concert, and I'm going to refer to Derek to talk a little bit about this.
Yeah, sure. So one of the things that is important statistically is that your measures are independent from each other, right? And there are a lot of elements of the genome and the methylation of the genome that are not independent, they aren't necessarily directly next to each other on the genome, but they, as Piyush said, move in concert. And so if you have a change in one area, you'll also have a change in another area almost every time, maybe occasional outlier, but this is evidence of a lack of independence. And so what we wanted to do was cluster our features so that we did have independent features in our feature vector. The other piece there that I think you mentioned in terms of the pre-processing combat piece was we had a list of a number of those individual CPGs or individual well sites on the chip that just didn't work very well.
Mm-hmm.
And so we first went down from 450,000, and I forget the size of that exclusion, but we got from Illumina a set of array locations that they actually didn't recommend using. And then all of this further processing is after having filtered out those poorly performing array locations.
Yeah, exactly. I think there was a study done in like 2011 where they identified cross-reactive probes that are prevalent in the 450K machines. And so we decided to just get rid of them and not worry about the effects, the unintended effects that they might have.
Yeah. So we've got our 320 CPG Island tokens, and that's our embedding for the space. Next, there's some pre-training going on. Tell me a little bit about that.
Yeah, yeah, and before we dive right into pre-training, let me just quickly outline the three main components of our transformer architecture. So, as Derek mentioned, first we've got pre-training, then we've got our mixture of experts architecture layer, and then we've got adaptive computation time. And so, in broad strokes, pre-training is where we on a high level, just throw all the data at it. We don't tell it what the problem is. We just throw all of the epigenetic data that we have at the model, and we tell it to learn the relationships between the CPG between the feature vectors. And for those of you who are very fluent in machine learning, the way we do this is by argminning the mean squared error. And essentially, it tries to recreate the data points over and over until it's able to achieve a fairly high accuracy in recreating it. And that's the pre-training step. And we found that the pre-training step by conducting ablation studies afterwards, we found that the pre-training step especially helped a lot. And we can pull up the exact number from our table. We can probably display it up here, but it was really quite significant. After that comes our mixture of experts architecture layer. So Derek, do you want to talk about that?
Yeah, you bet. And I'm going to actually pull up just to try and do this in real time. Stop editing. There is the ablation studies that Piyush was talking about. The pre-training was taking us from 91.9 up to the 97.1, and so that pre-training step really was quite essential in the overall accuracy that we were getting. So in terms of the mixture of experts model, I'm going to let, in just a minute, Piyush talk through the detailed block diagram that we've got here. But let me do just a brief discussion of what a mixture of experts model is before we go to that. Basically, what you're doing with a mixture of experts model is having multiple models run in parallel. So during training time, we haven't said what the specialty of each submodel is, but we have allowed it to train on the data and develop its own classification specialty just through the process of association in the data. And so then when we get an unknown sample, it is going to be sending it to this mixture of experts model and determining which of the models is actually providing the best classification. There's an example, an analogy to use here, and sorry if I'm stealing your thunder. You gave this in the other talk, Piyush, but that the mixture of experts model is kind of akin to what happens in the U.S. court system. So in the lower court, when a case is brought, the lawyers argue it, the lower court judges rule on it. If there's any points of law that need further elucidation, goes on up in the court system, eventually arriving at the Supreme Court. All of this earlier legal process can be thought of as our pre-processing and pre-training steps within our model. And when it arrives at the Supreme Court, there are lawyers that are presenting the case to a panel of 9 judges. And these 9 judges are ruling on the case as a single body. But generally, it's the case within the 9 judges that somebody will have a little bit better background or a little bit a more relevant training in a particular area, or just a bit more fluency with the arguments that are in front of the court at that particular point of time. Any of the judges could serve that role. It's not like any of them were trained to that particular specialty. But because of their background, someone will have the stronger voice, and they're generally the one that writes the final opinion for the court, or at least has an unweighted voice into that final opinion. The same thing is very true with the mixture of experts model, that we don't really track why there is a better background from one of the experts to the other, but in any given time, because we have multiple models running that have a little bit different experience and a little bit different basis for decision making, they're able to have an outsized weight on the final answer that is given. So that's kind of the rough picture of how a mixture of experts model works. There's a lot of subtleties in there, though, in terms of transformer models and just what is happening in the mixture of experts model. This is a weighted attention model, and so that weighs in terms of how that is working. And maybe, shall we go back to the block diagram?
Yeah, if you could share that. And for our audience, actually, the Supreme Court example for explaining mixture of experts came from Derek. We were talking right before the podcast on human life expectancy, and he came up with the original idea. And I stole it from him originally. He stole it back from me. So that's pretty funny. But let's talk about this block diagram. And I think, well, before starting on that, Derek might mention an excellent point, and that's that in our mixture of experts architecture, the specialists are specialized, but we don't know what they specialize in, nor did we train them to be specialized in any particular thing. That's just naturally how the experts work. They specialize in different domains. However, again, to emphasize, we did not train them to be good at like any particular thing. The idea behind this is like, let's first talk about why we're even using mixture of experts rather than just a core transformer. Anytime you're adding complexity, you must have justification for it, right? So there are 2 key benefits of having mixture of experts. The first is that you can increase the capacity of the model, the number of parameters, without increasing computation. So you can look at the number of parameters we've got in our model, but not all of those parameters are being activated at the same time all at once. That's the beauty of mixture of experts. Only a small subset of those are being activated. And this means that computation time is reduced. Another is that because we've got like such diverse and multi-cohort data from all of these different data sets, the mixture of experts architecture helps with analyzing and dynamically routing, dynamically selecting where each token goes, which expert each token is routed to. So that's what our gating network is used for in the mixture of experts step. So in the block diagram, we see this in step in the second column. The first column is just for the standard transformer encoder. So just token embeddings, then the multi-head self-attention, then the feed-forward neural network, which is repeated n times. Then we've got the MOE in the second panel, and then we've got another MOE in the third panel, and then adaptive computation time for the final panel. And maybe I'll push it to Derek to talk about ACT.
Sure. Yeah. So adaptive computation time is basically a trick to allow us to not have to compute as fully if the decision is obvious. So if there's a really tricky distinction to be made, we've got 2 classes that we're really not sure how to separate. Then it might be worth continuing to cycle around our mixture of experts model and trying to reprocess to get the best, finest distinction. But But if we you have a very clear distinction that on the first pass through it's obvious this is long COVID, then the adaptive computation time is essentially going to chop that additional processing off. And so it can be thought of as a computational trick to minimize the processing time. If we had decided to process to the full extent for every decision, we would have arrived at the same answer, but we would have used much more compute time in the process of getting there.
Yeah, that's right. And I was thinking about this a couple of days ago when GPT-5 launched, and this is kind of like the model router in the base GPT-5 version, where
Hmm.
if it's a simple question, it'll just go to the GPT-5 medium or small or mini. Um, but if it's a complex question, it might go to GPT-5 Thinking High or GPT-5 Pro. So for users who are familiar with that conversational engine, I think that's a good way of understanding how this works.
Yeah, it's interesting. The only thing that doesn't feel exactly aligned with that is that a lot of the task behavior and model routing is based on explicit elements of the input,
Hmm.
and so tool use is kind of picking out a piece of the problem task and finding the specialized tool that pertains to it.
Yeah, yeah.
And we don't really know a priori with the adaptive computation time what those tool use behavior equivalents are. It's more the results that come back and how confusable they are that allows the adaptive computation to continue.
Yeah, yeah, for sure. That's a good nuance to note. Yep.
Yeah.
Yeah, yeah. So we've covered pre-training, we've covered mixture of experts, we've covered adaptive computation time, and that's how our model works on a high level. And if you're more interested in like the specific, the numbers and the parameters that we've used, all of that is outlined in the paper. You can also take a look at our code base. We'll leave a link down to our GitHub and Hugging Face down below. Everything's open source, and we're happy to welcome new contributors and collaborators. And I think Derek might have a couple points to add there.
Yeah, sure. So one of the things, there are two directions that we really want to take Mobius. Direction number one is continued refinement of the ME/CFS long COVID separation. It is a model that is tuned to that and does seem to be the first step towards a clinical diagnostic that will be really important. But the other thing that we're excited about Mobius about is that it's applicable to so many other domains. And it basically, anytime that you can take a set of epigenetic data, and of course, we have the pipeline set for the Illumina data, but there are lots of different conditions that have an epigenetic element. So it's not just ME/CFS and long COVID that we can separate. We could separate whether if you're an ex-smoker and want to know, hey, have my epigenetics returned to something approximating a never smoker as an indication of my risk profile having gone down, or any number of other conditions that have an effect on the epigenome. And really, that's most things that we run into in life. It's quite interesting the number of subtle effects you can tease out of the epigenome. Even as simple a thing as changing a diet can reflect itself in epigenetic changes that then control the downstream processing through transcriptomics and protein production. And so by finding the original data sets that have these classes defined, in this case, people who had been diagnosed with long COVID or diagnosed with ME/CFS prior to 2019, if you can find those analogs in other data sets, then this application and this method will apply directly to it. And so we'd really like to encourage folks that do want to apply this to other domains to have a chat and talk about your data set and see if it will work for you. And finding other research domains for this to sit in, it'd be really gratifying. We also want to, of course, extend this to other data sets within ME/CFS and long COVID, and so are actively looking for partners and collaborators in those data sets to do so.
Yeah, yeah. Thanks for adding that. And finally, we are currently deep into pathways work, biological pathways, and we've got some evidence to support some stuff, but we're working on that right now, and we're very excited to keep everyone updated on our status.
Yeah, that one's really fun. Yeah, we'll have some updates here on pathway pieces pretty soon, but maybe we ought to give a bit on the fundamental idea in there, yeah, just as a
Mhm.
teaser for folks. Yeah, can you describe the multi-headed attention maps and what leads to the pathway pieces?
Yeah, so that's a great point, Dirk. So one of the things is we need to think about explainable AI and the interpretability of models. And um, so let's think of linear regression, right? It's a very simple technique, and that makes it very interpretable. We know exactly why it works under the hood. However, with more complex models like XGBoost and now with transformers, it's a black box essentially. Um, and so we don't know what's going on. But one of the benefits of the transformer architecture is it uses multi-head attention, and meaning we can create attention maps out of it. And using these attention maps, we can identify like the broad locations of these CPG sites. And using that, that makes this work to like 10 times more interesting because we can connect it very strongly to biological pathways and hopefully discover some new insights there.
Yeah. Yeah, that's neat. So we're working both on the pathways, which we have to validate because
Hmm.
epigenetics is only part of the story. After epigenetics, there's transcriptomics, which are the portion of the epigenome that is actually transcribed, and then there's protein production and actual biomarker assays. And we've got to actually walk down that whole pathway to validate that the epigenetic pieces that we're seeing are actually resulting in biomarker changes in the actual protein production. Before we can say that this epigenetic change is the root of some of these issues. At this point in the epigenetics, it's really just suggestive of a pathway rather than being diagnostic of a pathway. But after you do validate some of these pathway hints, it's really quite an opportunity because you do start to see what actual biological production has changed in the case of ME/CFS versus long COVID. And that tells you something very useful. From an empirical perspective, there's been a lot of work recently on using probiotics for the treatment of long COVID. And you get about a 2.2 odds ratio improvement in a 6-month treatment with bifidobacterium probiotic yeah, that is a bacterial species that is explicitly depleted in the case of long COVID, which is indicative that there are treatments, but that treatment doesn't work at all for ME/CFS, even though the underlying symptoms seem confusable. And so identifying pathways and identifying the root underlying biology of these post-viral illnesses is really important for developing treatment options and hints towards what direction people might go in treatment.
Yeah, yeah. And just like Derek mentioned, on top of epigenetics, there's so many more layers like transcriptomics, and we haven't gotten the chance to look at these layers yet. But that's one area that we hope to explore next. One thing I want to note is that there's a really cool paper that just came out a couple of weeks ago called Biomap AI, and they explored many of the things that we wanted to explore with this paper, and we're going to continue to work on. So we'd love to link down to that, and we would recommend that you guys take a read. It's truly a really cool paper.
Yeah. Well, that's great. That gave us a chance to dive a little bit deeper into the underpinnings in the computer science in this podcast than we did in the HLE podcast. I think with Tom, we talked a little bit more about the biology than we did here, and we're probably getting some viewers from that HLE podcast. If you found us directly, you may want to go to the HLE podcast, and we'll put a link to that in our show notes as well. Can you think of anything else we haven't mentioned, Piyush?
No, I mean we mentioned that we have our GitHub Hugging Face link below, and we encourage people to watch the HLE podcast if you guys haven't already. And we'll leave Derek and my emails down below, so feel free to contact us regarding collaboration opportunities.
Excellent. Well, thanks so much for listening, and thanks for co-hosting today, Piyush.
Thanks, Derek. This was fun.
See you soon.